Hello class, we are back in the lab today and we are going to be learning about motility of bacteria. So I'm going to go over a little bit about bacteria, how they move, what they use to move, and then we're going to go over a few different techniques, and then we are going to do what is called a motility stab. So starting out to talk about motility of bacteria, about 50% of bacteria are motile, meaning they have the ability to move, and they use what is called a flagella to move. And so flagella is an organelle that is attached to the bacteria, and sometimes there's one of them, or it can be several of them, two of them, a pair of them on either side. And they basically, when they are reacting to something, they are going to move it like a propeller, and this is going to propel them forward. Then they stop moving the flagella and they tumble until they stop. So it's not a very direct motion, and this is called the run and tumble method. And this is how the bacteria move. Now they're going to be moving in response to something. So there are three different types of movement depending on what the bacteria are responding to. Number one, there's chemotaxis. Chemotaxis is the movement in response to some type of chemical. We have phototaxis, which is movement in response to some type of light source. And then we have aerotaxis, which is movement in response to some type of gas. So those are the three different types of movement depending on what the bacteria is reacting to. So finding out the motility of the bacteria is very important whenever we are trying to figure out what type of bacteria we're dealing with. So this test is done or used whenever we are trying to identify an unknown. Also, motility stabs, which is what we're going to do today, can be used to keep bacteria for 2 to 3 weeks in the cooler. So it's a way for us to save our bacterial cultures also. So the few different techniques that your book mentions are a flagella stain. So this is where you stain the flagella, and then we look at it under the microscope, and if we see flagella, then that means that the bacteria is motile. We have to have a special mordant type stain fixative to be able to stain the flagella, and we don't have that, so we're not going to do that today. The other technique that's mentioned in your book is a wet mount, where you simply take a drop of broth and then put a cover slip on it and put it under the microscope. It's really hard to see the bacteria move with a light microscope, the ones that we have in our lab. So that's a reason why we don't do those. You also have a hanging drop, which is very similar to the wet mount, except for it's a little drop of liquid that you're looking at. There's a little indentation in the slide for your drop of broth to go, your suspension to go, and then you watch it under the microscope under the 40x magnification or objective lens. Again, with our light microscopes, it's kind of difficult to see the bacteria move. Also, a lot of times people will see what is called Brownian motion. And Brownian motion is just the natural movement of the water molecules. When the water molecules move around, they bump the bacteria, and it makes the bacteria look like it's vibrating. And a lot of people will think that that's true motion, but it's not. It's not a true motion where it's in response to something. It's just moving around because of the water molecules. So that's called false motion, and that's something that you want to be careful of whenever you're doing a wet mount or a hanging drop. The technique that we are going to do today, though, is called the motility stab, and we really like the motility stab in this class because it gives us more than just the motility of the bacteria. It also tells us a little bit about the bacteria's metabolism. So the motility stab is exactly what it sounds like. It is a stab. Okay, so we're going to use an inoculating needle to stab the bacteria. And motility agar is special because it has less agar in it than normal solid media does. Most solid media has 1.5% agar in it. And if you remember, agar is the solidifying agent. That's what makes the media hard. So it creates a matrix inside of the media. And so at 1.5% concentration, the bacteria cannot move through that media. Even if they have the ability to move, they can't because it's too dense, it's too thick. So motility agar has a third of that. It only has 0.5% agar in it. So it's more liquidy, so it's less dense. So it allows the bacteria to move through the media. Now, let's talk a little bit about what else we can see other than the motility. So what you do is you inoculate the stab, and then we read where the bacteria are growing at in the tube. And that's going to tell us, did the bacteria move away from where you originally placed it? Where did they move? Did they stay at the top where there's oxygen? Did they go all the way to the bottom where there's no oxygen? So that's the kind of things we're going to be looking for. For the results, there's 3 different results that we can get when we are looking at a motility stab. We can have a facultative motile organism, a facultative non-motile organism, and a aerobic motile organism. So let's talk about what the word facultative means. So a facultative anaerobe is, well, let's back up. What is an aerobe? So a strict aerobe is an organism who needs oxygen to survive. They have an electron transport chain. The only way that they can get energy is using that electron transport chain and then using oxygen as the terminal electron acceptor. So they have no other option, so they have to have oxygen. We are strict aerobes. Then there's a strict anaerobe. A strict anaerobe is an organism that cannot be live in the presence of oxygen. So if it's a strict anaerobe, then that means that oxygen could actually kill the organism. And then we have a facultative anaerobe. And a facultative anaerobe is an organism that can use oxygen as a terminal electron acceptor, but if there's not any oxygen present, it can do other things to get its ATP, to get its energy, like fermentation or anaerobic respiration, where it uses something else other than oxygen as the terminal electron acceptor. So facultative anaerobes are pretty cool because if there's oxygen available, it can use that. If there's not, then it says, okay, I'm going to die, I need to do something else, and it can switch the processes and switch to something else. So if the organism is a facultative motile organism, it's going to be growing at the top of the tube where there's oxygen, at the bottom of the tube where there's no oxygen, and then it's motile, so it's going to move away from that stab line. It's going to be able to grow other places in the tube. So basically, the entire tube is going to be cloudy. A facultative non-motile organism is going to be an organism that grows at the top of the tube, at the bottom of the tube, but it's non-motile. So it's only going to grow on the stab line. Okay? It doesn't have the ability to move away from that stab line. So you're going to basically see one straight line of bacteria going straight down the tube. And the last option is aerobic motile. And this organism has to have oxygen to survive, so it's only going to grow at the top of the tube. You're not going to have any growth at the bottom, but it is motile, so it can move away from that stab line. Okay, and so you're going to see growth just at the top, but it will cover the entire top of the tube. Okay, so now we're going to go over to the workbench. We're going to inoculate some motility stabs, and then we'll read the results. All right, so we are over here at the workbench, and I'm going to show you how to inoculate a motility stab. I have the results for 3 different bacteria to share with you, the 3 results that we went over, but I'm only going to show you the inoculation process on one, because there's no sense in me doing it 3 different times. So, of course, we want to use aseptic technique, and the tool that we're going to use to do this is an inoculating needle. So if you remember, we have an inoculating loop. We use that to do all of our stains and make our smears, and we're going to use the inoculating needle to do the stab, because we're stabbing it, so it would make sense we need to use a needle. So this is what our needle looks like. Okay, you want it to be as straight as possible, because when we're reading these results, we want our stab line to be really, really straight. So I've already straightened it, and of course we want to use aseptic technique, so we're going to use our fire boy. We're going to flame our needle, so you want to treat this just like if it were a loop. So we're going to flame our needle. And of course, you want to get that nice red hot color. And I like to flame the entire needle, okay, because you're going to be stabbing all the way down into the tube. And if I was doing this 3 different times with 3 different bacteria, you need to make sure that you flame in between each bacteria that you're doing. So, of course, we're going to let our needle cool a little bit because you don't want to go in with a hot needle because you're going to burn the bacteria. So we're going to let it cool just a little bit. And while it's cooling, I'll show you, this is my motility stab. So it looks like that it's broth, okay? But when you turn it upside down, it doesn't move. But you can see that it's kind of jiggly, okay? And that means that there's less agar there. So the bacteria has the ability to move, okay? So that's my stab. And so I'm just going to use my slant, okay, to get the bacteria. This is E. coli. So I'm simply going to glide across the top of the slant, just like that. It doesn't take a lot, okay? We're going to let this incubate for 24 hours, so the bacteria is going to grow and produce colonies. And so here's my stab, and I'm going to get in the middle as much as possible, and I'm going to stab straight down and come straight back up. And that's it. So all I did was take the bacteria off of the slant and put it into this stab so that it can grow for 24 hours. Now, it's important that when you're doing this that you get your stab line as straight as possible, and you want to go down all the way to the bottom and come all the way back up. Because we're wanting to see where the bacteria grows. Does it grow all the way at the bottom where there's no oxygen? The only way you're going to be able to see that is if you put the bacteria down there. Okay? So, now let's read our results because I have some that are ready to read. Okay, so here's my 3 tubes. This is my 3 different results. Okay, and so the first one that we're going to go over is the staph. Okay, so as you can see, there is a line, a white line of bacteria going straight down the center. Okay, so this bacteria would be facultative because it grew at the top where there's oxygen and at the bottom where there's no oxygen. And it would be non-motile because it didn't move away from my stab line. I've got my nice straight stab line. If my stab line was crooked, I might confuse that with motile because I might think that the bacteria moved away from the stab line. It didn't. I just suck at stabbing things. But my line is nice and straight, and that's what you want. Also, you can take the top off and look down in the center. It's kind of hard to see here, but there's a dot right in the center where the bacteria is growing. So it didn't move away. It might be easier to look at here. You can see it's right in the center, but it's not going side to side. Okay, so if you were to do an unknown and you saw this result, this would be facultative non-motile. The next, E. coli. You'll notice that the whole tube is cloudy. Okay? And the top, you see the top there is completely covered in white. It grew at the top where there's oxygen, at the bottom where there's no oxygen, and it moved away from the stab line. So E. coli is a facultative motile organism. Look at the difference. This is non-motile, this is motile. Do you see how cloudy this is? Okay, so both of these organisms are facultative, but staph does not have the ability to move. E. coli does. E. coli has a flagella. The last one that we have is Pseudomonas. So, you see that there's growth concentrated just around the top, but there is no growth at the bottom. The bottom is completely clear. There's nothing there. Okay? But there is growth all along the top of the tube. So, Pseudomonas is an aerobic motile organism. Okay? So, it has to have air. There's no growth down here. And it is motile. So, it moved away from the stab line. And you can see that by looking down, and you'll see that there's growth all along the top. Compare it to E. coli. You see how clear this bottom of this tube is and how cloudy this one is. So there's no growth at the bottom of this tube, meaning that this organism is a strict aerobe. So those are my 3 potential results for the motility stab. I've got a facultative and non-motile organism, a aerobic motile organism, and then a facultative motile organism. And those are the three potential results that you could get whenever you're doing a motility stab.